Guys, my name is Mr. Mpepu and today's date 18 September 2024 and the time there 33 minutes past 11. With that being said, guys, Anbonani, Dumelani, Avusheni, hello, guys, I personally welcome you to our life sciences revision classes term 1 week 3 today, protein synthesis. If you are new here, don't forget to subscribe, don't forget to hit that notification bell, and don't forget to like, don't forget to comment. If you are a returning student, guys, thank you for choosing in. Thank you for tuning in. Thank you for choosing me, and thank you for pushing this movement together. Yesterday we managed to do the RNA types and the locations. Right, today we'll be dealing with our protein synthesis, transcription, and translation in our 10 minutes so video. So let's go on. Yesterday we managed to cover up until here, and then today we'll be dealing with our protein synthesis. So let me do my timer and then we are on, guys. Protein synthesis. What is protein synthesis? Protein synthesis is the process in which the proteins are made, right? Let me do the pen. This is the process where the proteins are made. It's called protein synthesis, right? And then it's made by combining amino acid, which are the monomers for protein, and then we use the base triplet. We start with a base triplet of our DNA, right? Those three nitrogenous bases, one after another, and then this base triplet, they are going to form a codon, or they are going to be coded into a codon of mRNA. And then these mRNA codons, they are going to match with our tRNA complementary, which are called anticodons, right? So it will be 333. And then this process takes place in two stages, which is transcription, whereby a message is being coded from DNA into mRNA, and translation, whereby in this process takes place in our ribosomes, where in that message from mRNA is being translated to a protein using amino acid and tRNA and all those stuff. So we're going to do them today as our revision. But proteins are made up for linking various amino acids. As you know that we only have 20 different amino acids, and then these 20 different amino acids can be joined in a varied combination, guys, making a large or a long chain of more than 50 to 300 and more chains of amino acid forming one protein, right? And the number of amino acids are sequence determine the different types of amino acid. As you can see here, the bond between the amino acid, we have learned that it's called the peptide bond. As you can see, these are the amino acids that are being joined up until we join a long chain there. As you can see, but the smallest protein contain 50 amino acid, and then the proteins general contain 300 or more amino acid, right? But three consecutive nitrogenous base of a DNA strand are called the base triplet, and this base triplet determine which amino acid will be placed into the protein, as well as the sequence in which the amino acid will be joined, right? So if it's CCC, is going to be to form an mRNA that will be GGG, and it's going to this mRNA is going to complement with tRNA that will be CCC, and then this CCC will carry up a specific amino acid. Let's go and explain about this when we're explaining translation and transcription. But the protein synthesis occurs in two stages, which is transcription and translation, right? A transcription, that's where a protein. Let's move on. As we can see, the transcription, this is the process whereby an mRNA is being formed, or it's being transcribed from DNA. Transcription just means that copying something as exactly as it is. Since it's a messenger RNA, then it's going to take that message to the ribosomes to produce a specific protein, right? So as you can see here, the DNA inside the nucleus, and then we can see the nucleopore, the mRNA after being produced. Same thing. The DNA is going to unwind. After unwinding, it's going to unzip. After unzipping, the base, the one strand of a DNA is going to work as a template, and then the complementary nucleotides from our nucleus, they're going to form mRNA, and then that mRNA, when it's fully produced, is going to leave via the nucleopore to the ribosomes for translation, right? As you can see here, they try to explain it, but the section of DNA double helix is going to unwind. After unwinding, it's going to unzip when we broke that weak hydrogen bond, and then one strand as a template, and then from there the DNA is used as a complementary strand for a messenger RNA. When this is done, this one is used to using a free RNA nucleotides in a nucleoplasm. The mRNA now contains the code for the protein which will be formed, and then three adjacent nitrogenous base of mRNA, they are called codons, right? We know about this, and these codons, they are going to complement with our anticodons in the tRNA, right? This code for a particular amino acid we know, and then the mRNA moves out of a nucleus through the nucleopore into the cytoplasm, where it attached to our ribosomes, where where protein is going to be synthesized there. And then from there we move to our next stage, which will be translation. Translation is taking place in our ribosomes. You can see that will be our ribosomes, and then our ribosome is moving into that direction, and then from there our tRNA, this tRNA with a specific amino acid, this will be amino acid, this will be the protein formation, this will be the amino acid joined together by our peptide bonds. And then what is happening is that our tRNA it's bringing that amino acid with the complementary. As you can see, this one is going to complement with this one is going to complement with this one is going to complement with this, and then this one is going to move into this position when this ribosome is moving forward. And then from there, after this amino acid being joined with this amino acid, this tRNA is going to leave, and then it's going to collect another amino acid up until all this mRNA is translated, right? So we can move on. But the transfer RNA in the cytoplasm has 3 adjacent nitrogenous bases called the anticodons, right? We have the codons, we have the anticodons, we have the base triplets. And then our mRNA codon will complementary with the tRNA anticodons. Each tRNA will carry a specific amino acid according to the codons of mRNA. The tRNA will bring the required amino acid to the ribosomes, right? So we understand this. The amino acids are linked by a peptide bond to form a required protein. This is the process that is happening in a one cell, right? This is the cell that is happening. From there, the mRNA is going to be produced. Then the mRNA is going to leave via the nuclear pore to our ribosomes, and then ribosome is going to translate that mRNA into a specific protein, right? And then, but note, it's very important to know the difference between the base triplet, that will be the DNA. So this one we can have CTA, and then the codons mRNA, that will be GAU, and then from the anticodon, that will be CUA, right? So let's make sure that we understand these three. But the effect of mutation in protein structure, if we have the mutation is the change in the nitrogenous base sequence of a DNA, and then since mRNA is copied from the DNA molecule during transcription, this will result in the change in the codons, right? So if it was CTA, and then this T, it was replaced by A, the codon is going to change, and then after the codon change, as a result, the different tRNA molecule carrying the different amino acid will be required, right? And then the sequence of amino acid changes, resulting in the formation of different protein. If this anticodon here was bringing a leucine or something, it's going to bring another a different amino acid, right? But not in all conditions, because if the same amino acid is coded for, there will be no change in the protein, since a different amino acids can be carried with using different triplets or different tRNA. Sometimes we see that they say there was a mutation, and then after that mutation there's no change because the same amino acid is still at the place, right? So yes, the protein synthesis activity, but identify the process above. The process will be translation. As you can see, the ribosomes, the mRNA, the peptide bonds, the amino acid, and the tRNA, right? So you can see there by name, the organelle A, molecule B, the bond E, organelle A, ribosomes, molecule B, RNA, the bond E, peptide, provide the letter and the name of the molecule that carries the amino acid, that will be the tRNA, and then the monomer of protein, that will be amino acid, right? Name, they describe the process occurring in the nucleus which results in the formation of mRNA transcription. They just want transcription, right? According to the table, how many codons code for phenylalanine? Then you just come and count 1, 2 only. What is the anticodon of glutamic acid? Glutamic, as you can see here, they give us the mRNA codons, so they want the anticodons, which will be the tRNA will be CUC, right? So that will be the answer there. And then the section of mRNA is the following base sequence that is read from the left to the right, but give the DNA base triplet for the last codon of this section mRNA. Here it will be thymine, guanine, because it's DNA, right? And then but the first amino acid coded by this section of mRNA, GAU, you come here and say GA, this is GAU, right? GAU, yes, aspartate, and then you write aspartate, and then you are done, right? As you can see, straightforward, we are done with our protein synthesis. And then the code of life, end of topic exercises. You can come and do these exercises if you have time. As you can see, the molecule of RNA is copied from the DNA by the process of transcription, and then we move on. You can do this if you have time during your spare time. A DNA analysis found to contain 14% thymine. What the percentage of molecule of cytosine? This one 14 plus 14, and then the rest will be 36, if I'm not mistaken, right? So let's move on. The gene, and then 1.2 biological term, but the sugar, there's the sugar that is that is a component of DNA. This one will be deoxyribose, right? The oxyribose sugar, and the protein that form the part of chromosomes, histones, and then a segment of DNA coding for a particular characteristic gene, the bond that form between the nitrogenous bases, weak hydrogen bonds, so forth and so forth, right? So do these biological terms. And then column A with column B contain ribose sugar, DNA, discovery of DNA, no, location of DNA, nucleus, mitochondria, the process whereby one molecule produced two identical replication, pairing of nitrogenous base DNA. So you can do this in your spare time, guys. You can do this, name the, and then we give a diagram, name the nucleic acid DNA, and then push in 1, 3, 5, 2, 1 there will be the nucleotide, nitrogenous base 3, guanine, since this one is cytosine, and then molecule number 5, phosphate group, and then bond number 2, hydrogen bond, weak hydrogen bond, right? So you can do these activities in your spare time. Our 10 minutes, it's up. The diagram below represents the DNA replication, as you can see, and then from there identify the following. You can identify there, and then we move on to our section B, protein synthesis. This one, nitrogenous base, and then but write down the base. Our time is up. Write down the base sequence of the anticodon triplet one shown above. This one will be the they want anticodons, right? So make sure that you know or you understand this terminology here. The codons, anticodons is tRNA. So here you have to provide mRNA, which will be uracil, adenine, uracil, cytosine, and then anticodon will be uracil, adenine, guan. Hey, this one is guanine, this one is cytosine, right? Hey, mistake. Hey, let's move on. But you see here, right, you understand this one. We have to find mRNA, which will be adenine, uracil, guanine, and then the anticodon tRNA will be uracil, adenine, cytosine, something like that, right? So we move on. The table below shows the amino acid that correspond with the different mRNA codon. Give the correct sequence of amino acid for DNA triplets 1 up to 3. Then as we already have the AUG, it will be methionine number 1, and then number 2 TCT will be AGA, something like this, arginine, and then number 3 there CCGGA, something like this, and then this one will be glycine. So practice this, guys, practice this, and then draw the RNA nucleotide with the complementary base adenine. They just want you to understand that adenine is going to form with a uracil, right? So this one, how you draw it, it's up to you, and then we'll see, you have to see that sugar, and then we have to see that phosphate group, right? So we move on. The diagram below represent the process of protein synthesis. Study them and answer the questions that follow. And then as you can see that what is happening here, this one will be our mRNA, this one will be our DNA single strand or a template strand. This one is mRNA because you have uracil, right? Well, identify the structures, label 1, 2, 3, DNA strand, and then number 2 will be tRNA, number 3 will be our ribosomes, right? So my time is up, guys. Let's move on. You can do this on your spare time. Number 3, the diagram below represent 2 stages of protein synthesis, transcription and translation, and provide the labels for molecule 1, where is molecule 1 DNA, organelle 6, ribosomes, and then this one will be the codons, this one will be the what we call tRNA, this one will be the amino acid, this one will be the strand, and then number 3 here will be the nucleus, if I'm not mistaken, right? Mistake. So let's move on. State the difference between DNA and RNA. You know, deoxyribose, ribose, thymine, uracil, and then the third one, they only want 2. And then from there, how many of the following are coded in the DNA template sequence above? Nitrogenous base, you count them, and then different types of tRNA molecules that are required to form the polypeptide form with the piece 1, 2, 3, 4, 5, 6, 7, right? So we are moving on. Use the table below, determine the amino acid sequence, and then we have the 4 and 6. They want 4 and 6. So for you to get this 4 and 6, since it's DNA, you have to come with mRNA and also the anticodon. The number 4 it will be adenine, cytosine, uracil, right? And then number 6 will be guanine, adenine, and then guanine, right? And then we go and check with your anticodon here. G will be C, this one here will be glutamine, number 6, this one here, and then this one will be U, we'll start with UGA will be 309, right? You don't have to know the amino acids, you just have to know that if I can code this the name, they will provide the name either way. You know about this. But if the T in the 6, the question is done, we are done, guys. Let's meet tomorrow under meiosis. Thank you guys. Peace until we meet again tomorrow. My name is Mr. Pepu. Peace. Peace.